Hello, everyone. Today, I will present to you an analysis of tumor necrosis factor alpha induces human bronchial fibroblasts to produce cytokines, implications for airway inflammation. This research was jointly conducted by the research group led by Professor Wang Xiaozhi from Binzhou Medical University Hospital and the research group by Professor Dong Liang from the First Affiliated Hospital of Shandong First Medical University, and was published in the Med Research Journal. Fibroblasts regulate the progression and resolution of pulmonary inflammation cascades through multiple mechanisms and have recognized functions in many processes. Su et al. Uncovered that adventitial stromal cells and myofibroblasts recruit pro- and anti-inflammatory immune cells in allergic airway inflammation. Malay et al. Interleukin, IL-17 was elevated in asthmatic airways and induced human bronchial fibroblasts to produce IL-6 and IL-11. Which promoted the progression of airway inflammation. Osei et al. Reported that IL-1α/β induced the production of granulocyte macrophage colony stimulating factor, thymic stromal lymphopoietin, TSLP, IL-6, and IL-8 and inhibited the production of collagen, fibronectin, and periostin. Drake et al. Revealed that TNF-α significantly upregulates the mRNA expression and protein secretion of IL-6, IL-8, CC motif chemokine 5, CCL5, and TSLP in lung fibroblasts. However, as a subtype of lung fibroblasts, how bronchial fibroblasts respond to TNF-α stimulation and subsequently regulate immune responses remains unclear. To further verify the role of TNF-α-induced bronchial fibroblasts in inflamed airway with asthma, human bronchial fibroblasts were stimulated with recombinant protein TNF-α. And gene changes were analyzed through gene expression profile sequencing. This study found that 711 genes were significantly upregulated, 608 genes were significantly downregulated. The DEGs further analyzed and found that the inflammatory cytokines and chemokines were expressed, such as IL-6, IL-1β, IL-15, TNF, chemokine, CX3C motif, ligand 1, CX3CL1, death-associated proteins 2, DAPK2, TSLP, CCL2, CCL5, CCL7, chemokine, CXC motif, ligand 1, CXCL1, CXCL2, CXCL3, CXCL5, and CXCL6. Interestingly, the expression levels of fibrosis-related genes collagen type 3 alpha 1 chain, COL3A1, and collagen type 4 alpha 3 chain, COL4A3, remained unchanged. In contrast, fibronectin 1, FN1, collagen type I alpha 1 chain, COL1, collagen type I alpha 2 chain, COL1A2, and actin alpha 2, ACTA2, exhibited increased expression, while transforming growth factor beta 1, TGF beta 1, and VIM exhibited decreased expression, albeit with a fold change of less than 2. Gene ontology, GO, enrichment pathways based on the background of the upregulated DEGs showed that TNF-α-induced bronchial fibroblasts are closely associated with the programmed cell necrosis signaling, T helper 2, Th2, cytokines production, eosinophils, neutrophils, and so on. Then, Western blot showed that the expression levels of IL-1β and TNF-α in TNF-α-induced bronchial fibroblasts considerably increased. The expression levels of alpha-smooth muscle actin, alpha-SMA, was not changed, and the expression levels of fibronectin, COL1A1, and TGF-beta1 were substantially decreased. The ELISA results showed that CCL2, CCL5, CCL7, TSLP, CXCL1, CXCL2, IL-6, and IL-1β levels were considerably increased by TNF-α-induced bronchial fibroblasts in the supernatant. These results further highlight the importance of TNF-α-induced bronchial fibroblasts in mediating airway inflammatory responses in asthma. In summary, the uniqueness and innovation of this study lie in its use of human bronchial fibroblasts and transcriptomic approaches to systematically characterize genome-wide changes. The inflammatory phenotype of bronchial fibroblasts may provide therapeutic insights into asthma-related airway inflammation. We welcome citation study. Thank you all for listening.